Now, I said at the beginning of this chapter, every living thing on this planet, every group, has at least one virus that can infect it. All plants, all animals, all fungi, all prokaryotes. So we're going to be discussing at the last part here of section 5.3 are viruses that infect bacteria. Turns out their mechanisms are quite a bit different than what we've previously talked about here in this section, which were for animal viruses. Viruses that infect bacteria are commonly referred to as bacteriophages. Phage means virus. Bacteriophage means bacterial virus. These are viruses that can only infect bacteria. We're going to see that for all intents and purposes, these are going to be double-stranded DNA viruses. Are there exceptions? Probably, but none we're going to worry about. Different bacteriophages can infect different species of bacteria. There's no one virus for all, just like we talked about with host range and tropism. For animal viruses, it turns out there is species-specific viruses. You look above my head, you can see the generalized steps when it comes to bacteriophage infection: attachment, penetration, replication, assembly, release. Pretty much the same thing as we talked about before. Here, big difference is the shape of our virus. The shape of our virus is going to be completely different. You look over here, you see this lunar lander looking thing. The nucleocapsid composing a head, it's got a sheath, and it's got the legs, or things that looks like legs. It comes, it lands on the surface, and then those leg-like things contract down, jamming the sheath, the tube, through the membrane, or membranes, whether you're talking gram-positive or gram-negative, through the peptidoglycan cell wall into the cytoplasm, where you get release of the viral DNA. Viral proteins never enter the cell. Only thing to enter the bacterial cell, viral DNA. Now, once in, two pathways can be followed here: lytic and lysogenic. Lytic, as we just saw a few slides ago. Viral DNA enters in, host cell machinery, DNA polymerase, RNA polymerase, ribosomes, begin reading, copying, and making more viral proteins, viral DNA. Assembles, self-assembles, and you get enough of them self-assembled inside the bacterial cell, and then the cell pops, releasing thousands. One goes in, thousands come out. Or you get what's referred to as the lysogenic cycle, or lysogeny. The lysogenic cycle is the bacterial version of our latent infection. The viral DNA enters into the cell, and then it becomes a prophage, quite similar to the provirus we saw with the animal viruses. Viral DNA inserts into the circular bacterial genome, and then it stays there. Every 20, 30, 60 minutes, however long it takes for the bacterial cell to divide, 1 becomes 2, 2 becomes 4, the normal course of the cell cycle for a bacteria. All new cells that are being made will carry the prophage. Some signal occurs telling the prophage to pop out of the genome, and then it suddenly starts acting like it would if it was the lytic cycle. That viral genome gets read by the two different types of polymerases. Ribosomes come in, make new viral proteins. Everything self-assembles, cell pops. You look at this picture here. This figure that I've borrowed from another textbook kind of shows you what it's talking about. One side you have the lytic cycle, the other side you have the lysogenic. One's not better than the other, it's just the final outcome could be slightly different. The lytic cycle One bacteria gets infected, one virus goes in, hundreds, thousands, or thousands come out. Done. On the flip side, with the lysogenic cycle, say it's E. coli. E. coli divides every 30 minutes as long as there's nutrients there. Virus infects, prophage inserts, and 24 hours later, prophage exits out, gets the signal to exit out. One cell was infected, but because 1 becomes 2, 2 becomes 4, 4 becomes 8, and so on and so forth, 24 hours later, you now have 1 million bacteria, all with prophages. In my mind, lytic is down and dirty. Get in, get out as quickly as possible. Lysogenic, that's, the long view. Wait it out. Because not all those million of E. coli are going to suddenly start popping because the phages are releasing. Only some of them. The ones that didn't, because the prophage remained, they will continue on being a bacterial cell just with something hidden in it. And what we find is that because of lysogeny, the prophage inserting and the prophage coming out, it's not always cut and dry. It's not always all clean. Sometimes when the prophage inserts, it inserts next to, a gene of interest. Given time, that prophage will then pop out of. I say pop out. There's other things happening that makes it occur, but we're just going to go with pops out of the bacterial genome. And it pulls along with it part of the bacterial genome. That part could be part of, or most of, or all of a gene for antibiotic resistance, a gene for a new toxin, a gene for some new ability. Next bacteria that gets infected by that phage now is going to have that ability as that prophage inserts into its genome. It's a gene's a gene. Doesn't matter whether it comes, from one bacteria or another. So that's always a concern for us microbiologists.